My name is Rachel Richardson, and I'm here to talk to you guys a little bit about this multi-omics pipeline automation via multi-omics analysis portal that we've been working on this last fiscal year. So we're going to start really basic. What is multi-omics? As most of you guys are probably aware, if you have a biological system, there's all sorts of data that you can pull from that system. Recent advances in technology have allowed us to pull multiple different kinds of data from the same biological sample. And this has been really, really useful. Examples of this has been in microbiomes, soil or gut. If you look at patients, before and after treatments is often very helpful for that. And under any kind of experimental condition that you're interested in these samples, you can also use multi-omics for. But why do we care about multi-omics? So when we're looking at this biological system, if we only look at one omic, we only see one part of the picture. If we get more omics involved, we can see a greater picture, right? This elephant example is a pretty good metaphor, but if you're thinking in a more scientific sense, one of the things you might consider is phenotype. There's a lot of individual variability in phenotype, and a lot of times that regulation happens not just on one omic level, but multiple omics levels that we're interested in seeing. Now, conducting a multi-omics analysis can be very involved, very many steps to that process. This is a typical workflow for doing the multi-omics analysis. And just to kind of walk us through it, the first thing you really want is kind of what question are you trying to address, right? And try to design an experiment that will address that question well. After that, you have to actually collect your samples. You have to run your instruments. You have to annotate or ID any of the molecules that are coming off your instruments. And then you have to assign a relative abundance to that particular biomolecule. And after that, you have to figure out what tests are valid for that, what visualizations make sense, and any other sort of statistical analysis that makes sense for the question you want to answer. So there's a lot of steps there, and there's a lot of expertise involved in all of that, right? So if you think about the experimental design as well as statistical tests, that requires some statistical expertise, obviously. If you think about the sample collection, running the instruments, and doing those annotations, that requires some of that biological and chemical knowledge. Lastly, these data sets tend to be very large. I think most of you might have some experience with that, but actually moving them around into different applications or pipelines, you can do a lot of formatting, and a lot of different computer science expertise is required to do this effectively. So our big goal with this project was to essentially create an application that removes that burden on our researchers, right? So we want to enable fast and easy access to your data exploration to get to your conclusions faster and empower you guys to do it in a valid and reproducible way. So our first attempt at doing this was an application called PMART. PMART was originally designed for bottom-up proteomics, both label-free and isobaric labeled. And it was so successful in that process that we expanded it to lipidomic and metabolomic data sets. So all of these are based on mass spectrometry data. And as you guys might be aware, mass spectrometry data can be difficult to handle sometimes, right? You'll see a molecule in one sample, but maybe not in a couple other samples, creating blanks essentially in your data. And in addition, if you have something like peptide data, you're really more interested in the protein sometimes that peptide is associated with. And having that allocation is something that takes some time and expertise to know how to do. PMART handles these kind of data anomalies that are with the mass spec instrument. So in addition, it handles data transformation, data filtering, outlier detection, normalization, as well as statistical analysis. It does it very well. So I'm going to take you through a couple more points in our actual application setup that helps us get back to those goals of valid and reproducible data. So one of the big things in having this in an application like this is that every single step can have a visualization. Everybody wants to know what's going on with their data at every step of the way. So this kind of visualization is very helpful for our users. In addition, not only can you download every plot in every table, but you can also download a report at the end of the application to tell you exactly what you did so you can reproduce the process, right? Lastly, there's guidance available throughout the applications that give you either a little bit of additional information about your results or about the analysis being conducted. And further, there's some functionality within the application itself that supports very data-specific options for our researchers. So this tool in particular is Spans. And what Spans is doing is it's helping the user pick the best normalization method that's introducing the least amount of bias into their data. And as you can imagine, this can be very data set specific. This kind of functionality is well supported in our applications across the board. So PMOD-R has been very successful for its uses, but we are aware that there are other things that people might want in their multi-omics processing pipelines. For example, if you have data that is not innately supported by PMOD-R, for example, FTICR, you might need completely different processing steps. You might want to look at multiple omics types at once and see what kind of integration, what similarities are happening between those omics data. And maybe you want to see one biomolecule in the Krebs cycle, or 10 or 50 at once, but not the whole summary statistic. That is all different functionality that is not innately supported in PMAR. We thought about it, but we decided against it. And I will tell you guys why. If you guys have ever walked into a restaurant and they hand you a menu, either one of two things happens. They hand you a book that has everything they've ever cooked ever, or they hand you one that's very specific for the meal of the day, right? So as you can imagine, going through a book of a menu and finding the right thing for that time of day and what you're actually wanting out of your meal, that's a much longer process, right? In comparison, a single page finding exactly what you want is much easier. The same thing for our applications. If there's too much functionality in one application, there's too many buttons, you need a manual just to use the application. In addition, if you think back to the menu as well, if you wanted to update that menu, to update a whole book is a much more tedious process of finding where you actually need to update the book, and actually printing it all out is much more difficult than if you only would have one page. Very much for the application space as well, finding the very specific features you need to change is much easier when the application is smaller. So we decided to instead make a multi-omics analysis portal, a one-stop shop for our multi-omics analysis. So essentially what this portal does is it will host a number of applications to meet all of our multi-omics needs. Back to the menu example, it's essentially having a separate menu for each desired outcome of your multi-omics analysis. Kind of just looking at this video here, it's going through a couple of the different filters we have for the different applications. So our analysis goals is one of the great ones, so very specific to what you actually are trying to achieve with your data sets. In addition, you can filter by which data types are appropriate, or if you have data uploaded into the application, it will tell you specifically what's appropriate for that data. Talking about what is supported in MAP right now, we saw those three, but there's a multitude of other ones, just to pick out a couple. So we talked a little bit about PMART. In addition, we have IPMART, which is similar, but handles that integrated aspect of multiple omics types. In addition, we have MODE, which will let you look at 1, 10, 50 different biomolecules in very granular detail. And I would like to also hit the point that MAP is helping you pick these applications appropriate to your data. In addition, it will help you pick the applications in the correct order. So like restaurant menus, first you have dinner, and then you have dessert, very much so. I'd also like to point out that MODE and NITO are also on today's agenda for Flash Talks. Please stick around for David and Logan's talks in a little bit to hear more. So just to give you guys an idea of what MAP actually looks like, we have a short demo here. The first step, we obviously need to upload our data into the application. Right now, we support direct upload through CSVs, but we're also working with the Nexus team to be able to directly access any data generated from EMSL user proposals. after you do all your uploads, you can access different applications and just click on it. And what the app will do, it will seamlessly load all of your data into that application. This particular one is PMART, right? And we saw some of that earlier. And once you're done with your analyses that happen with that particular application, you can export it. Just name it a very specific file name and bring it back into that map application. This data that you're using in your processing is saved every step of the way. And when you want to use a different application, for example, this is mode that will be coming up later in the Flash talks, it will automatically load that data into it, and you'll be able to visualize very quickly. Lastly, just to talk a little bit about how we did this, because we're very proud of our work. There was some front-end development and back-end development, front-end being all of the interfacing that you saw today. That was mostly done in Shiny R packages, such as Tidyverse, as well as some JavaScript and CSS to make it pretty. The back-end development is pretty great. It's mostly designed to keep our application's integrity, as well as be able to move between applications without having to do a lot of formatting and processing that's typically in other pipelines. If you guys have additional questions, please let us know about that. But in the meantime, we'll just wrap it up, kind of recapping the functionality of MAP. It is an automated software to assist users and give that expertise when going through every step of these processes. And we also really value the validity of all the analyses that are happening, as well as the reproducibility of these analyses. So you can incorporate it directly into your publications that you want to work on. In addition, that effortless moving between applications is very valuable to us, knowing some of the data science that happens to kind of reformat data. And lastly, we've got some robust back-end support to move the different softwares into that portal and support additional softwares as they become available. So our MAP anticipated release date is January 2023. We need beta users. If there's anyone interested, please contact us. In addition, if you have software that you would like to see in this kind of a portal, please contact us as well. We are looking for additional applications. Thank you guys so much for listening. If you want to contact us, please talk to either Lisa Bramer or Kelly Stratton. Their emails are up there, pretty straightforward. But yeah, thanks. I'll take questions now.
Thank you, Rachel. Great talk. Do we have any questions, please, for Rachel?
Yes, hi.
Oh, great. Okay, we have one online first. Yes.
Okay, it's great to know about it, Rachel. I have a kind of a crazy question. We have an old data set that was taken almost a decade ago at PNNL, proteomics data set that we like to examine for some different aspects that we were not even thinking about at the time. And as far as I know, it's in your repository up there. And so my question is, can you do an old data set? Can I pull that out and dust it off and run it through PMART?
Yeah, so if it's in the Nexus repository, we should be able to support it. We do support a very specific data file, right? So as long as you have your identifiers for each sample and a relative abundance associated with that, it should work well within many of the applications that we already have in the MAP portal.
Okay, do I need to apply for a user grant to do that?
Yeah, I would touch base with either Lisa or Kelly, and they should be able to set you up with what the necessary needs are for that. It'd be great to have that data set to work with and check out if everything's working well for that.
Okay. Thank you.
Yeah, thank you. Anything else?
Yeah, we have another question here.
So for the implementation of the correlational test, the ANOVA, Kruskal-Wallis in R, I assume, with MAP, how do you mentioned that you're able to cover missing data issues, so on, that come up a lot in mass spec. How do you implement that in R, given the difficulties of correlation with missing data?
So those aren't the only tests that you can necessarily run. with the statistical analysis. For example, there is the G test, which looks for the independence of missing data, which can be very useful when you have a lot of data missing. There are minimum requirements. If you wanted to run an ANOVA, for example, you'd have to have at least 2 observations in each of your group to actually run that test. So we're not doing any sort of imputation just because there's some debate on what makes the most sense for different data sets, right? Or at least in some of the current applications we have. That's not to say that there's not future applications that will specialize in that.
Understood. Thank you.
Is there any other question? Yes, we have more.
I'm trying to do things on the fly here. So to look, I don't know, is any of this available right now? Like, and is it available through Nexus? Is that where you go in to find these tools?
Not yet. We do have some local versions we're using for our development. And as a beta user, if you wanted to check out some of this application, these application spaces, I would definitely encourage you to get in touch with Lisa or Kelly. And we'd be happy to share some of what we currently have with you guys for your processing needs.
And then the follow-up is there, so do you just have R packages that you've wrapped in some way, or can you just go in and code in R in this framework? And load in some new package and run it?
Yeah, so currently all of our applications are UIs. They require no coding whatsoever. We try to make it as easy as possible for our researchers. If you wanted to integrate a new application, you could go do that coding. But yes, a lot of them are based on packages that already exist in R in that space for actually running some of these analyses.
Can't actually run R.
No, not from the application.